Welcome, everybody, to our Innovators in Analytical Chemistry Education seminar for June. Before I introduce today's speaker, I just want to mention that our July webinar will be moved up a week. So instead of being on the last Thursday of July, it'll be on the second to last Thursday of July. And I'll put a link to the registration for that in the chat. And actually, July's speaker is here with us today. So Shannon Acetone from Trent University is going to talk to us about teaching analytical chemistry from a mastery perspective. If you have signed up for our subscription, you don't need to register. You'll automatically get the link the day before. But if you haven't and you'd like to register, the link's in the chat. I've also put a link to the paper that our speaker will be mentioning today in the chat for you. So with that, on behalf of my co-organizer, Bhavik Patel from the University of Brighton, who's having some emergency dental work done, I'd like to welcome today's speaker from the University of Bern, Gunnar Schwarz, who's going to be telling us about a survey of quantitative analysis experiments so we can start thinking about what people are doing in the lab and what we might want to be doing as we make our plans for next year. So welcome, Gunnar. Take it away.
Yes. Thank you very much. Also for the invitation from the organizers. I'd like to welcome you all to my presentation in this webinar series. I will, as mentioned, mainly focus on one of my latest publications, but I will also provide an additional discussion of the data and then switch to another publication which mentions an experiment, and I will compare the findings of the first to the second. So as seems to be usual for this webinar series, I will take a very few slides to introduce myself to you. My academic journey so far has not been long distance-wise. I was born, raised, and educated mainly in Berlin. I also studied chemistry there. And this then was followed up by a doctorate in analytical chemistry, which I did in the group of Professor Michael Lindscheid. There I researched on quantification of proteins with mass spectrometry. Afterwards, I moved to Switzerland, Zurich, where I joined as a postdoc Professor Detlef Günther's group at ETH Zurich, and there I researched with and on laser ablation ICP-MS. And 2 years ago, I moved to Bern to join the University of Bern as a lecturer, where I am now also an independent researcher. I always had interest in education, teaching, and during my last year of my diploma studies, I already started as a tutor for a lecture course, and this then went on during my doctoral studies as a teaching assistant for both lab and lecture courses. And during that time, during my doctoral studies, I had the opportunity to develop a new lab experiment because my then supervisor said we should use our ICP-MS in one of the lab courses. And then it was kind of up to me, or I took on the challenge to develop a lab experiment. And that's that I based on my research. So the students in that lab course quantified proteins from an egg with ICP-MS by coupling lanthanides to those proteins and then compared it to a Bradford assay. Afterwards, I kept on publishing a little bit. I described the summer camp, which was organized by the group, and back at those days ran for 20 or so years. And then as a postdoctoral student and at ETH Zurich, I described our approach of engaging the public during an outreach event where we analyzed qualitatively objects from the public like jewelry or coins with an ICP-MS right on site. So not in the labs, but in a kind of a tent outside the main building of the university. From the very beginning of my, say, teaching career, I had the hypothesis that teaching is often driven by questions. Okay, I was a tutor then, so what do tutors do? They ask questions, they put problems to the students. So questions were always at the center of my considerations for teaching. And when I had the opportunity, then given by Professor Detlef Günther at ETH Zurich, to kind of pursue my own career there and develop my own projects, I immediately went on to develop multiple choice questions for classroom response systems. And that was my my own publication. Here, dismissing the tool analogy for these classroom response systems and replacing that with an interface model. And the second publication was more concerned than on the questions themselves, question development. That was also kind of a starting point for this. And because one of the reviewers for this first publication kind of criticized me for complaining that there are not enough multiple choice questions for this area, then I jumped on it and compiled all the multiple choice questions I developed and published those then in a third publication. So this publication contains in the supplementary information over 250 items of multiple choice questions, and then also discusses the development guidelines for those questions not in terms of summative assessment, meaning exams, but more like for an exercise purpose. Okay, I want to switch now to our main topic here, meaning the lab experiments for quantitative analytical experiments. To me, chemistry is an experimental science, which mainly takes part is done in the lab. So there are a lot of lab courses and hand-on experiments for students studying chemistry or related subjects like biochemistry, pharmacy. Even our medical students have some chemistry labs here in this university. So, and the labs are very nice to develop diverse skill sets, which you can also find and discuss in the literature. But from my perspective, quantitative analytical experiments are, with my background, the main focus. And as I will discuss later, I couldn't find anything that resembles an overview of what is done typically. So I took on this task to survey the literature on which experiments are described, what analytical experiment aspects are covered in the literature and which not, and how are these experiments framed for teaching. So what are the circumstances? How are those experiments also presented to the students? For such a literature survey, one needs a scope. Otherwise, one gets easily distracted from contributions which are related, but not the main part of the focus. So I limited my survey to experiments described from 2014 to 24. That those are 11 years, just has to do with my move from Zurich to Bern. When I arrived here, I stopped the survey a little bit, and then another year was gone, so I included 2024 as well. I used only contributions for the experiments from the Journal of Chemical Education, because this journal provides or has a certain structure which enables easier comparison. There are, of course, other journals publishing such experiments. You can find some here, but they are more irregular in their publication frequency, and they don't have that kind of a common standard, which is then a little bit difficult for comparison. But you can find other experiments as well. The focus was clearly on quantitative analysis and specifically not just a yield determination after product synthesis. So I excluded that, and I excluded demonstrations and simulations of experiments. I also didn't want to cover pH determinations or articles on entire lab courses, which then deal more with how to organize a lab course. And I didn't include instrument development or characterizations of such instruments. For that, I mainly did a manual search of the online journal issues based on titles and abstracts, and that resulted then in 161 contributions. And because a few papers had multiple experiments, those resulted in 164 experiments overall. And while reading all these papers, I took notes, many notes, and then assigned categories to analytical and pedagogical aspects of the experiment. And this is the list of these aspects, and I will go through all the aspects and categories in a second. I also want to mention here already that from my perspective, the analytical and the pedagogical parts should run in parallel. So there's not one in front of the other or in different directions. I will pick up on that picture later on. And I would also put in front the limitations here. There were some serious judgments called necessary. So not every exclusion or inclusion of a paper or a category assignment was straightforward. This may not seem obvious at for some categories, like, I don't know, group size, but there were always some issues with some papers where I had to decide on my own. It's just that, and that's how it characterized. So there may be a little, from some examples, you may have another opinion. Yeah, okay, but I think overall the results would not be different. And I did not check, for example, achieved students' learning outcome with stated learning outcomes. I didn't cross-check categories. I also did not have insights just from the papers regarding pre-lab activities, costs, or the supervision. I will also comment on that a little bit later. There was no investigation of study programs or trends over time, say more titrations in the last 5 years compared to the first 5 years. I didn't do that, but if you're interested in that, data is available and you can easily do that on your own if you're interested in a particular change, for example. No consideration of use of statistics for communicating the results. That would have interested me. I tried that, but I couldn't find a way to really deal with this. But could be if somebody else finds something there, would be interesting to me as well. And the results do not necessarily represent general lab course usage. That's why I didn't investigate trends over time, because it's not representative of what's actually going on in the labs, I think. And I did not consider the evaluations. How are they done, or what are the results of these for these experiments? Because typically evaluations are a requirement for publishing an experiment in this journal. So first and perhaps foremost, the result is that I have now a huge list which is available as supporting information with this publication. It consists of, as I mentioned, 164 experiments. It's searchable and it's more detailed than what I will show you, of course. For example, here you have sample preparation, a sample preparation column, and here acid digestion and dilution, and you will find this in different categories in the results I will show you later. So there are more details on category assignments also found here in this file. And there's even an abbreviated version of this list, which I think is more of an overview for easier search. This is the intention, and my hope is that other instructors for lab courses get some inspiration from this list without anything that I will tell you later. Or secondly, that they find that one of their very interesting experiments might be worth publishing as well. So maybe you can look through the list and then decide, oh, I have something that has not been covered yet for some reason. And you may be motivated to publish then an experiment on your own. So now I will go through the aspects and comment some highlights and perhaps some difficulties in category assignment. First of all, the sample. So the analytical aspects I sorted kind of in order of the analytical procedure steps. Here we can immediately see that food and beverages are very common samples. And together with water samples from the environment, meaning rivers, lakes, this is the most popular sample that is analyzed. On the second, we have consumer products, anything you can buy essentially at a supermarket, including over-the-counter drugs. Those are also included here. The third category, I named that academic samples. And what I mean by that is pure analyte in a solvent. Later, after publishing the paper, I thought, okay, that may be a little bit misleading here, academic sample. So you can understand it as a mock sample or artificial sample. This is just to contrast with the other authentic samples, which are used in a different way and are not just prepared for the sake of analysis. This is the meaning behind this term. But I said mock or artificial sample may be better understandable than academic sample. Unusual examples of these samples are banknotes, fingerprints, sewage sludge, and some experiments, only some actually, incentivize the students to bring your own sample, like a water bottle or a chocolate or something. Analyte. You can see the organic compounds cover half of all the experiments. Some analytes were explicitly proxies. Best example, I think, were airborne particles for viruses for the corona pandemic. During the corona pandemic, somebody with the students researched on how these viruses distribute in air, and so they approximated this by airborne particles. Very, very interesting also for that time frame. Then the element category also included ionic species. Otherwise, we have inorganic anions, ammonium, inorganic compounds, and here I separated even metal-containing organic compounds from the organics. There were only a few cases of speciation, meaning different oxidation states or forms of complexes. And there was also, surprisingly for me, no case of quantitative determination of an isotope ratio or distinguishing between isotopes. Isotopes were used for one quantification, so based on isotope standards, but not a determination of an analyte based on that. Sample preparation-wise, not all those sample preparations were actually carried out by the students. I will comment on that later. And I subsumed many common operations and procedures under the term basic operation. So this involves that because once I separated them in like a graphic diagram that was more or less meaningless, so all the more common procedures I subsumed here, and you can see that also covered a lot of experiments. That's why extractions, derivatization, digestions were less common. 11 experiments had no sample preparation because they used the samples directly without any dilution, for example. Measuring instrumentation. This distribution more or less follows what other surveys had found in practicing quantitative analysis in industry or education programs. If you consider analysis in general, so not quantitative, but also what is in the sample, structural elucidation, then mass spectrometry and NMR becomes more prominent. But for quantitative analysis, this is the typical what you typically see also with other surveys. Mass spectrometry is somehow hidden here in between in the chromatography, liquid chromatography, and gas chromatography. It plays, of course, a role here. But this is somehow hidden here, and because I'm from when I present this at conferences, the vendors from mass spectrometry companies always ask me, "And where's mass spectrometry?" That's why I'm telling you that here. And I subsumed spectrophotometry and colorimetry already in this category because makes to me less sense to separate them. Then quantification, most of the quantification was done by external standards. Both external and standard edition can be combined with internal standards, and you also have a category on internal standards only. There was only a limited use of certified standard reference materials. Of course, they're costly, yes, but they are common practice in analytical chemistry. So why not use that? Also show that in a student experiment. It doesn't need to be the best quality and most expensive thing. It could also be something you prepare on your own in the lab, but it often was just not shown as a possibility. Now switching to the other side, the teaching side, because this is more or less, not completely, but more or less independent on what the analytical side does. Yeah, as mentioned, it's not completely independent, but to a certain extent. First of all, the student's academic level. So experiments were not limited. This is a kind of a disclaimer to students of chemistry. There are many study programs involved, including medical students, pharmacy, geology, environmental scientists. So a lot of study programs had these experiments, and I didn't record which study programs were involved. But from the distribution, you can see there are many more experiments in the upper division, undergrad, say third year students, compared to first or second year. I think this somehow reflects when in the curriculum quantitative analysis is taught, perhaps even with a time delay, because at many institutions there is a lecture first and then a lab course in a later semester. But this can also run in parallel. That's why there are not many experiments on a graduate level. I saw now in the last year and this year a little bit more in this journal, but this may be just because I'm now recognizing that a little bit more or easier. Could be. A word on learning objectives. I based my category assignment on this very neat publication, but you can also find alternatives in the literature. So this is not the last word on learning objectives or differentiation between learning objectives. This is also a kind of a concept in science education. And if one is not used to that, that comes with certain difficulties. I just want to mention that the assignment of constructs is not a fixed thing. For example, you could associate problem solving with numerical calculations, say calculation of a pH in a certain solution. This could be problem solving, but that's not what is meant. Likewise, for students to devise an analytical strategy for a given analytical problem, say how to prepare a sample, which instrument to use, and how to prepare or how to analyze the result. could be a problem, could be problem solving, or could be experiment design. I use the experiment design for this approach, but you may think it's different. The description in this paper is also not very clear on that, but this is not because the authors are not clear, the literature is not clear on that. So I assigned when students were incentivized to develop an experiment, I put this into the experimental competencies category. I will show you later, this makes not much difference to the results here. Be surprised in a few slides. So this is the learning objectives category, the distribution. So you can clearly see it is dominated by experimental competencies and disciplinary learning, and much less higher order thinking, affective domain. Especially the higher order thinking skill section surprised me a little bit, because this is somehow in the education literature, science education literature, pushed a lot, but I was not able to see that. I only based the category assignment on what was specifically stated in the contributions text. So I didn't try to read into it. This may be an issue because maybe something was intended and then not explicitly mentioned. Could also be. I've somehow the impression that this was at least sometimes the case. There was a very wide variety on how the learning objectives were stated or phrased in the text, from extremely generic to very specific, like almost like a description of what the students did stepwise. So this was a little bit complicated to assign. But now we come to what was the most surprising for me after compiling all the results into these graphics here. The experimental procedure. So in the literature, you can find kind of a demand or recommendation that students should be involved in developing the experiment. Whether you call it open inquiry or whatever, it doesn't matter to me here. This is just to contrast when an experimental procedure is provided, like it was the case for 84% of all the experiments. I found only a few experiments partially provided the procedures. So like some sections were missing and students had to fill it in, or it was not as detailed as necessary for conducting the experiments, or not provided at all. This, as I mentioned, contrasts with the recommendations in the education literature a lot. This is not a criticism from my side. I'm not advocating for one over the other, like with all the other experiments or all the other categories I show you here. I'm just stating it as I found it. And maybe something of a discussion point later on. Because there may be reasons for that. We can think about that. So group size. The majority of experiments involved groups of students working on the same experiment. The median group size was 2.5. I assigned generally mean values. So if the paper states 3 or 4 students in a group, I assigned the value of 3.5. There's also in contrast with a low number of mentions of transversal skills, because these transversal skills are usually associated with collaboration skill development, communication also between the students and not just reporting stuff. So I think from my interpretation of this finding is that the group work typically involved in these experiments was not based on a learning objective, but organization reasons, too many students, too few instruments. Yeah, this is not a criticism from my side to the instructors or authors. It's just statement of findings. But on the other hand, often it is not clear whether the tasks are actually performed together or the workload is divided for the different students within a group. This is how I found group work, despite the number of students, can also be a positive influence on the experiment if the workload is actually divided. Okay, I will show you later. Time-wise, median value was that students spent 4 hours in the lab. The range was from 1 hour to 24 hours. Of course, 24 hours in different sections and not consecutively. This mainly excluded pre- and post-lab activities, but that depends then on the text of the contributions, whether this was separated or just put into one time mention. Sometimes authors mention specific measures of time savings, like not letting students prepare the sample or the standards. Student activities. Not all students did all the analytical steps themselves, as I just mentioned. So sampling, in the sampling step, only very few experiments involved students in sampling. It's only like 9%. And parameter optimization is another aspect which is not done by the students typically. Because that, again, is a time thing. It's a time-saving mechanism to provide parameter optimizations. And there was this very special case I like to mention. The instructor incentivized the students to earn a driver's license for a flame AAS, which I found very interesting because I did the same some years earlier. For other reasons, it was really cool to see, and especially seeing this term driver's license for an instrument. Good. Otherwise, they are well covered: sample preparation and standard preparation, and essentially all the experiments involved the students in the measurements. There were some exceptions when there was just automated measurements. Okay. Almost done here. Assessment is dominated by a written report or post-lab questions. Not always was I able to distinguish between, because the students were asked to provide answers to the post-lab questions within the written reports. Other assessment types are very rare, including accuracy. So whether or not students were able to provide an accurate result, accurate analysis, only 4% of all the experiments. That was also a little bit, yeah, I don't say disappointing, but surprising, because quantitative analysis is typically, the aim is to provide accurate results. And should students learn that or not, and should be then also assessed on that? Yes, discuss. I want to mention a few perceived shortcomings. Sometimes there was just missing context, no specific or vague analytical questions to be answered, or sample and analyte are often selected by instrumentation and not the instrumentation based on sample or analyte. This is understandable in many cases. I also mentioned the lack of isotope or isotope ratio quantification, chemometrics, automation, details of data processing, determinations and handling of result uncertainty seems to be lacking in those contributions. And on the more of the teaching side, the learning objectives, where if you really systematically read those, you really see there are as a wide range of quality, say. Student developing an analytical procedure is clearly missing. If this is a stated learning objective, generally for quantitative analysis, this should also be included. Sampling, students are rarely included during the sampling activity, but the basis of a good analysis is the sampling. Is working in group effective for individual learning or sharing workloads? Question mark. Depends highly on how it is actually done in the lab, and not all the contributions actually elaborate on that. And yeah, many procedures, many experiments were really streamlined. So it's the cookbook analogy fits here very nice. So they were not challenged by some kind of deliberate obstacles. So something did not work. If something did not work, it was usually the instrument's fault or lack of maintaining or something. Yeah, and then data gathered often remains unused to answer an analytical question. It's just sometimes the impression of mine was it's analysis for the analysis sake and not for getting an answer to a problem. Also mentioned the last point. So I want to invoke this picture here to best that the teaching and analytical part run in parallel, not one in front of the other or in different directions, especially when you consider a third party, namely students. So this is how it should look like. Students should get the best of both worlds. So from depending on who is who, analysis and the teaching, and not these other two possibilities. And I will briefly discuss this on these slides here. So why do these choices, these categories are kind of choices of instructors, why do they matter? So again, I'm not advocating for one over the other. In any case, it's just a thing that instructors, developers of experiments should consider. So authentic samples may be good for assessing students on their ability to provide accurate results, because when you have academic samples, of course, are that. If you have authentic samples, you can motivate the students, you can really bring so-called real samples into the lab. But if you want to assess students on their analytical performance, the academic samples or mock samples may be a better fit, because then you know what's inside and how much in this case. Do you use an instrument based on an analytical problem, or do you select an instrument based on availability? It is in the lab and the curriculum. Both are fine, but if you, for example, have an analytical problem you want the students to solve, then it's kind of up to you to then check that the instrument is also available. My idea of such a lab course was always to have one sample, per and multiple instruments, I don't know, like an apple, and then analyze the apple with the different instruments, that the students are not associating, say, water samples with a certain kind of instrument. On the other hand, if you have one sample per instrument, you show more variety, okay, of samples. Yeah. And common equipment versus uncommon special in-house built equipment. That was a little bit tension. I saw there's some tension in those contributions. This depends again on what you want the students to learn. If you want to familiarize the students with common equipment they perhaps use in the careers, the common equipment is the way to go. And if you want to train the students to use more uncommon, to be able to think into that new system, unusual equipment, then you can use anyway a different approach. More on the teaching side. So I also already commented a little bit on group work. I think both can work. For analytical experiments, it's also my experience that groups are more often involved than in synthesis labs. But individual work is where the students will end up anyway. So wherever they work, they will conduct the experiments on their own. So that should be not forgotten. Of course, for analytic chemistry, typically the instruments are more costly, and then it's better to have two per group or something. Then I found a nice way of workaround for parameter optimizations. Don't let the students optimize the parameters themselves, but as a time saver, this publication by Sidner and all, they provided the results of this parameter optimization. And this then incentivizes students, or then asks the students to pick the parameter based on this presented graphs or whatever it is. I found that a very nice idea. You can share data between the students to do some statistical analysis perhaps, or don't share the data. Provide experimental procedure or open experimentation, I already commented on that. And then assessment based on accuracy or based on reports and data analysis quality. This is tension between the possibilities. Maybe a last comment here on this. So I'm not advocating against reports, but as an instructor, I would think about, does one more lab report really improve the student's ability to write lab reports? Yes or no? If the answer is yes, then by all means ask for lab reports. But one should always think about alternative approaches, even modifying the requirement of a lab report. I will show you an example. So first conclusion here, some ordinary experiment may be a better fit than a special or exceptional experiment. Because when I take the most common categories, then the experiment is as follows. An organic analyte, typically ethanol, in a food or beverage sample is prepared by dilution and measured by UV vis spectroscopy, absorption, and calibrated with external standard. And this experiment is carried out by 2 students in the 3rd year, and that because they should develop experimental skills and disciplinary knowledge. The experimental procedure is provided, and they spend 2.5 to 4 hours in lower lab. And as an assessment, they write a written report with post-lab questions. This is at least familiar to me as an experiment, maybe from your studies or even from your practice as an instructor. This is, kind of a common experiment. And what I want to point out here is that just mentioning these categories here and characterizing an experiment in this way doesn't tell you if this is meaningful for learning for the students, by there are still aspects which are not communicated with these categories and by the papers, right? For example, the quality and quantity of supervision which takes in the lab with the students cannot be as such communicated here. So this is kind of a disclaimer here. We can characterize these experiments with these categories, but it doesn't show you a full picture on is it a good experiment for teaching. So still, few recommendations. Make the context and relevance of performing an experiment explicit. No analysis is carried out for its own sake in the industry or research. It always has a purpose. It always has context. More emphasis on translation from a general problem to an analytical question, because that is challenging for students. It can be simple. It can be just orally with the students in a pre-lab discussion, but it should be there. And the general question then should be answered based on the results. Always let students consider alternative procedures. Can be brief, can be in a written lab report right after the experiment, or how could you improve now the precision of the experiments if you want to do that again, like that. So ideally, that is kind of the take-home message here. Explicitly justify each analytical and pedagogical choice for a fitting approach of the experiment. Lastly, I want to compare that with an experiment I also published right after this literature survey was online. This was also the reason I undertook the literature survey, because during the development of this experiment and then writing the manuscript, I noticed the lack of an overview on experiments. So that was the motivating factor here, or the occasion, say, to undertake the study. So To give you very brief context here, spring 2023, we replaced Flame-AES, an old instrument, with a new one, and this was the occasion to redesign the experiment. Previously, that was the determination of water hardness and tap or bottled water, which then moved into the lecture part. And so we had an opening for a new experiment. It was an analytical lab course for second-year students, groups of 2 or 3 students, 2 groups each week. And there was a preceding lecture course. This is very important. I will tell you in a second. There was a preceding lecture course where all the basics of quantitative analysis were given to the students and also exercised with a related task. Also, as a given was the lab time of 4 hours. So not more, not less, I mean, plus minus 30 minutes or whatever, but 4 hours was kind of the target. And we were able to purchase a new instrument, which is abbreviated MICAP-OES. It's easy to understand because it's just the same principle as an ICP-OES, but the plasma runs with much cheaper nitrogen instead of argon. So especially for teaching context, maybe a very attractive option. Multi-element detection capability, compare that to general Flame-AES setups. And for most elements, similar or better detection compared to Flame-AES, sometimes even close to ICP-OES. But this was the instrument. It's also a very compact instrument. Just from my experience, I can really recommend looking at that. Very interesting for teaching. At least for teaching. I'm not that experienced to comment on the analytical performance for real routine applications, but for teaching, definitely something to look at. So we had the objectives to use the knowledge and skill we already provided to students with the lecture course to then require from the students to diverse a strategy. So we did not provide any procedure, any experimental procedure. We just gave them a problem, and then during these 4 hours, they were tasked to develop an approach and then conduct the analysis and prepare a written report then afterwards. Students were allowed to do different processes, like just dissolving these tablets, these dietary supplement tablets in water, filter them, for example, and or performing acid-digestion. And typically they did both. We really required them to prepare the samples differently to then being able to compare. So that was the regular samples. Students were also allowed to bring their own samples if they asked first permission from the teaching assistant. And I was first a little bit skeptical that they can fill that or perform that experiment within the 4 hours, but that went very well, I have to say. That was very, very good by the students. So this is the characterization, kind of done in a manner that I have shown to you. The key point is here, we did not provide any procedure for the students, but they still, based on the lecture course, were able to do that. And this was kind of the aim, the major aim anyway, that they learn how to do that as with an example. And I wanted to comment briefly on the written lab report. It was not a full report, for example. I said, okay, they will not learn much when they write, say, 2 pages on ICP-OES or MICAP-OES. So I tasked them to briefly summarize that in a maximum of a half a page, so that I can then read through that and find the main mistakes they make, misconceptions. And that was the second part. They should use the introduction to give me 2 examples from the literature of ICP-OES applications, so that they were incentivized to go into the literature, find something, and then summarize this paper by 1 or 2 sentences. And this was, I think that was a teaching opportunity, very valuable teaching opportunity. I did not extend on it in this publication, but you can also tweak the written lab report a little bit, that it doesn't become just another report. That's my message here. So finally, I want to acknowledge my former supervisor, Dr. Günther, for really giving me opportunity over 10 years to develop my own career path in a way. And then my co-author, Chiara Fabretti, from the described experiment, and others who helped me to improve the manuscripts. And last but not least, I want to thank you all and the committee again for inviting me. And I'm open for questions and any comments and discussions.
Great. Questions for Gunnar? Yeah, Chuck, go ahead.
Thanks, Gunnar. Thanks for not bringing up the soccer game either.
Detlef Günther is so disappointed when he will see the YouTube recordings that I didn't mention soccer in any way.
I was interested in the fact that only 4% of the experiments looked at accuracy. That is now I see the experiments were a little bit biased to third year.
Yes.
But do you have a sense of, like the actual labs performed by universities? Is it that low that focus on
I would think it's higher. I have no idea what they're doing. That is something that really would interest me, like a survey. And I have that on my to-do list, kind of.
Okay, great.
Whether or not I'm doing it is another question, but it's really interesting. So I think there are more experiments than are covered here. There are also other aspects. I mentioned that in the publication. That, for example, when you would extend the scope of the survey, then you would easily include, for example, more experiments where the procedure is not given. So I think that and the accuracy thing, the assessment on accuracy, is a matter of what are you looking at. So these are then lab courses, lab experiments which are more established and then are considered not worth a publication, perhaps, because they are based on titrations, gravimetry, and so on. So more classical approaches. But I would welcome every publication that makes, for example, the case. You can also do that. We find a way to do that with instrumental quantitative analysis, like FLAME-OES, ICP-OES, whatever. You can think about even electrochemical instrumentation. If that requires then a certain kind of sample precision and so on. That would be really interesting because titrations are different kind of thing.
Okay, thanks.
Yeah. Yeah, Chuck again.
Yeah, sorry, I'm trying to give people other time. I was surprised. I think slide 13, you showed the different types of experiments.
Yes.
And I was shocked at how high the atomic spectroscopy was.
Yeah.
Yeah. Yeah, that may be just because, like I know maybe only 40% of instructors use that chapter in my book.
Okay, okay. Yeah.
And I guess though, yeah, it's again, you're looking more at third year experiments. Maybe that's again why the bias is there.
Yes, that can be. I think in general, again, titration could be even higher if you could consider or if those experiments would be published from first year courses, not on the topic of quantitative analysis, but just general chemistry. If you then I would think titration would be even higher or a higher rate. Could be. I have not done that, but yeah, I would think that. And that's why it's a little bit biased against that.
Okay, that makes sense. Okay.
I'm curious about when you found experiments where students were optimizing parameters, was there more emphasis on optimizing sample preparation or instrument settings, or is it a mix?
Um, it's more a matter of instrument settings, I think. But I would have to look that up again. I would think there were instances where students considered and even tested potential sources of interferences. So that could also fall into that category in the end. Um, uh, One of the drawbacks of the mentioned MICAP instrument is that it takes about 20, 30 seconds sometimes, depending on the parameters again, to get a data point. So parameter optimization is a very slow process with this instrument. That doesn't diminish the capability of then doing an analysis. But if you use it then for students to optimize a parameter, say gas flows, for example, then this becomes a time-consuming task. What we had actually a few groups who did that and did that very well because they asked for it, or it was part of the discussion, and then decided that this could be part of the experiment. So another argument here from my side is there is a paper. I'm very bad with names, so sorry for anybody who I cannot remember. But there are, I think, at least two papers in the Journal of Chemical Education which were published in the last, say, few years on a simulator of ICP-MS. And I immediately saw when I saw the publication and I tried this simulator software that it would really not be nice to combine this because it really simulates an actual existing instrument to use a pre-lab for the simulator, then optimizing to parameters, and then go use the time with the instrument in the lab, and then all the argon consumption to really perform the experiment. And by the way, when I was just reading through the paper again, I thought, yeah, this is would be some very interesting study to really have a case where a simulator, whether it is virtual or with virtual glasses or whatever, to do some parts pre-lab, pre-real lab, and then have a real lab afterwards. And then also show similar learning gains and actual cost savings, because simulators are not cost neutral. Some cost really money, depending on how sophisticated it is. So that would be really interesting to study, actually. Or have somebody and then see the results. That's always the easiest.
Yeah, thank you. And thanks, Chuck. Chuck has found the papers, hopefully, that you were mentioning and dropped them into the chat.
Yeah,
I was
exactly.
just
Yeah. Amy, yeah.
I was thinking it's nice to have the students optimize the sample preparation because often that is the part that needs a lot of specific fine tuning for an analysis. But sometimes it's a little scary because they might end up with a sample that you don't really want to put through the instruments.
Yes,
It's
exactly.
tricky.
Exactly. Yeah. And the same goes with sampling. I mean, sampling is so important, and I think from what I see in, say, textbooks or curricula, sampling is not always part of the education anymore. But it's so important that the knowledge is not just with the sampling experts, but actually also with those who are actually running the experiments. And then, for example, showing in an experiment the impact of sampling. They are very good descriptions. Also in the textbook by Harris and Chuck, really cool examples. However, it rarely translates into the lab. I mean, the lab is typically not the locality where the sampling is taking place. But whenever you can have students taking a sample, whatever it is, it could be just a water sample from a river. But even taking a river water sample is not as straightforward as it sometimes seems.
Great. Well, we're out of time, but I'm sure Gunnar would be pleased if anyone wanted to reach out with additional comments or questions, and I'll be sending out the recording probably tomorrow. Thanks so much, Gunnar, for coming and speaking with us today. Really interesting work.
Yeah, thank you. And
And
thank
as
you
a
for the
reminder, I
invitation.
hope I'll see you all in July for Shannon's talk.
See you then.